Good evening, everyone, and good morning to you. My name is Suzanne Barnett, and I'm hosting your webinar for you today. Now, before we get started into the webinar, as usual, I would like you to just give me a show of hands to make sure that you can hear me first before we start the presentation. So click on your hand button for me. Thank you, Scott. Thank you, Micheline. Andrea, thank you. Okay, if you can't find your hand button, you can always put yes in the question box for me. That would be great. Okay. Super, thank you, Donna. Thank you. So I'm assuming that the rest of you can hear me. And my advice to you is to switch off any applications that you've got on in the background, because it does improve the quality of your webinar throughout the presentation. Now, before we get started, I really want to sincerely apologise to those of you in Australia. We did mess up with the timings because the clocks changed in America. We messed up with our timings here and we took you back 1 hour as opposed to forward 1 hour. So today, obviously you got the email to let you know that we were starting at 12:30, and for all of the future webinars, we've got 4 of them left, we'll also be starting at 12:30 in the afternoon. We anticipate that this will take no longer than an hour and a half today, so sit back, get your pens ready and start making some notes. We will allocate, as usual, some time for questions at the end. So today we're on our presentation 7, which is GABA and Immunoglobulins. So today, once again, our speakers are Sterling Hill, Carolyn Ladowski and Cynthia Smith. And the first part of today will be presented by Carolyn.
Thanks, Suzanne. Okay, so welcome everyone. Sorry again for the mix-up with the time, but thanks for joining us. We are going to be discussing the glutamate and GABA pathways. And this section might look small, but for me, and I know certainly for Cynthia and Sterling, it's a really significant one, and probably one that I find the most interesting. The whole glutamate and GABA synthesis issues with GAD and glutamate dehydrogenase is really key to controlling someone's excitatory neurotransmitter behaviour. And we're really going to focus on that, particularly in light of what we discussed last week when we were talking about viruses and bacteria upsetting the glutamate to GABA balance. So that's really key. And then we're going to talk about immunoglobulins. And Cynthia will take you through the IgG and IgA, and I will take the IgG and IgE, and I'll take you through the IgA. So let's look at glutamate and GABA first. And I think it's often overlooked. And if we look at the work of Amy Yasko, she's looked into this pathway of excitotoxin damage, and she believes that it really is key to our neurological inflammation. And she's described this mostly with kids in autism, and she explains that excess excitotoxins definitely cause an imbalance in the flow of calcium, and that in turn leads to activation of a very complex, inflammatory cascade. And it's the release of these inflammatory mediators that ultimately are responsible for the cell death of neurons, and that's what we talked about the other day. And glutamate, as we know, is our main excitatory neurotransmitter. It's our precursor to GABA, and we know that GABA is our inhibitory, or one of our key inhibitory neurotransmitters. And both glutamine and glutamate we still need. They're still important in human physiology because they not only help with tissue pH buffering, so they have a very key role in maintaining our acid-base balance. They're crucial in supply of gluconeogenesis, particularly in the kidneys. They help us with our ammonia removal. So not only does the nitric oxide synthase pathway, but these two are key in working with that pathway in getting rid of our ammonia. They obviously help as well with glutathione and most importantly, I think, with neurotransmitter function. So if we have a look, GABA is obviously the major inhibitory neurotransmitter in the brain. It's synthesized, as you can see here, primarily from glutamate through the glutamic acid decarboxylase, which is a marker for GABAergic neurons. And GABA is transported in amino acid transporters. You can see those there. They're called VIAATs, and they're transported to the synaptic cleft, and that's where it interacts with our GABA receptors. So you can see there GABA-R is our GABA receptors. And GABA can activate then our ligand-gated ion channels and G-protein coupled receptors. And it's this mechanism of action that then has an interaction with our glial cells in our brain. And so our first enzymatic, breakdown of GABA involves that GABA-T or GABA transaminase. And really importantly, and I want you to remember this aspect, is it has to use alpha-ketoglutarate from the Krebs cycle to regenerate a molecule of glutamate for every molecule of GABA that is catabolized. So that's a really important point, because when we look at our citric acid cycle and our urea cycle, invariably I see people with very low alpha-ketoglutarate. And so straight away we know that this is going to be having an effect on their cycle. And so what's also important to remember is that it's our pools of GABA and glutamate, they're constantly being replenished. So if we don't have enough alpha-ketoglutarate or things like that, we're not going to be able to constantly get this supply. And this GABA-T reaction that you can see then goes on to form succinate. And we know that succinate is a really important component, again, of the Krebs cycle where alpha-ketoglutarate is formed. So it's this ongoing conversion of glutamate to GABA and back to glutamate, and that's what we call our GABA shunt. So it's this constant reforming of glutamate to GABA. And also our major precursor of glutamate, glutamine, is synthesized in our glial cells. And that's transported to neurons where glutamate and then GABA are metabolized. And GABA undergoes a sort of a reuptake in the glia and is then converted to glutamine. And then again, we have our GABA shuttle. So it is quite a complicated system, but I guess the key takeout is that we need to have good levels of both, but not too high, definitely, of glutamate or glutamine. And I think that's really key. This is a really great paper, and I would highly recommend everyone read it, because it is probably one of the most comprehensive explanations of glutamate metabolism disorders I've ever read. And so if we now know that glutamate, glutamine, and GABA are converted, let's look at what happens when, you've got these genetic issues going on. And you can see here that glutamine and glutamate they're converted to and fro. It's certainly not a one-directional flow. As I've just said, glutamine, glutamate are constantly they're going forward and backwards. And glutamine not only its function includes protein synthesis, because we need nitrogen for the synthesis of amino acids, purines, pyrimidines, nucleotides, and also for the transport of amino nitrogens to our intestines, our liver, and the kidney. So it plays roles as not only a precursor, but also an inhibitor of nitric oxide production. And a large percentage of what we ingest in the way of glutamine goes towards our energy production by enterocytes. And that's huge. We all know that glutamine is so important. But it has, I guess, the key role or advantages that this has for the enterocytes is that it really means that if we supplement with glutamine, it actually may be completely ineffective in raising our plasma glutamine levels. So if we look at this a bit more closely, when amino acids are broken down in the liver, glutamic acid is produced and nitrogen or ammonia. And this is then transformed into urea by the urea cycle. And as we've just said, alpha-ketoglutarate then can be used to produce energy via our citric acid cycle. And glutamate will take the nitrogen away from our exercising muscle, and it does that again with the help of alpha-ketoglutarate. So you can see that the glutamine is really involved in the detoxification of ammonia, and if we have an acidic condition, then ammonia is released. And acute acidosis, the research tells us, stimulates renal cell uptake and metabolism of glutamine. If we have chronic acidemia, it's associated with low glutamine, because what it does is increases the flow of glutamine from the liver to the kidney, where the ammonium ion is removed, and so it lowers our pH. So acidosis really stimulates glutamine uptake in the kidney and tends to produce very low plasma glutamine. So you would expect to see sort of signs of acidosis, perhaps an elevated anion gap on our bloods. And if we look at this GAD gene there, you can see that it is crucial to convert glutamate to GABA. The immune system is also influenced by moderate variations of glutamate, and we sometimes see a high fasting glutamate as a factor causing even low lymphocyte activity and low T4 counts. And this ties back in with what we were talking about with our kynurenine pathway the other day, where we said that viruses, infections cause this disturbance in the picolinate pathway, the kynurenine pathway, and that activates our NMDA receptors and influences our glutamate levels. So symptoms of hyperammonia we might see as headache, irritability, fatigue, mental confusion, poor concentration, food intolerance reactions, particularly to high protein foods, because when we eat proteins we're going to be creating ammonia, and really a low protein, high complex carb diet with say alpha-ketoglutarate, vitamin B6 and our branched chain amino acids may actually be really good in helping to correct this, particularly if our glutamine is high. And we all know that glutamine is so widely used, sometimes up to 10 to 20 grams a day, but really oral glutamine is not generally effective for modifying our plasma glutamine, and that's because of our conversion of glutamine to ammonia and glutamic acids. Some researchers say, well, we really shouldn't be giving glutamine at all. So I think this is a really, and certainly with blood sugar regulation, this is really important. And you can see how important the genes are. And so you can see the GDH obviously creates alpha-ketoglutarate that then goes into the citric acid cycle. So this is a really key. We just think GAD is the only thing that we're looking at here in terms of being able to create GABA, but we're actually talking about many, many other important processes where glutamine and glutamate is being affected. So I'm going to hand you over to Sterling now, who's going to take us through these GAD genes. Just to our GAD, glutamic acid decarboxylase, there's actually two GAD enzymes, which are GAD65 and GAD67. So GAD67 is encoded by this gene, GAD1. So when you see the research, GAD67 relates to this particular one, GAD1, and GAD65 is encoded by the GAD2 gene. Okay, so over to you, Sterling.
Hi. All right. The GAD gene is crucial in breaking down glutamate, an excitotoxin, into GABA, a calming neurotransmitter. Its cofactor is P5P. So if P5P is low and or lysine is low, you will see elevated glutamate and low GABA in most cases. This leads to anxiety, agoraphobia, panic attacks, panic disorder, and can cause more issues with PTSD. Also, when P5P is low and you see elevated glutamate and low GABA, also know that the body will be making endogenous oxalates, which will compete with sulfation. And that in turn will shut down methionine synthase, also shut down the Krebs cycle. This is why the GAD gene is related to cancers, Alzheimer's, Parkinson's, schizophrenia, autism, multiple sclerosis, type 2 diabetes, pyroluria, and many other diseases. GAD is related to alcoholism and heroin addiction. Alcohol and heroin temporarily make GABA. There has been a link to alcoholism and MS. Alcoholics tend to hide behind the bottle because it calms them greatly. There is higher increase in MS in alcoholics. For those of you in the United States, doctors are prescribing fluoroquinolones like candy. I know most of you in the U.S. know this by now, and we're seeing a lot of the damage come out on the news and everything. They're actually, people are this week going up to Washington, D.C. to black box it a few more times. But right now, the FDA has black box fluoroquinolones when given with NSAIDs and steroids. NSAIDs are non-steroidal anti-inflammatory drugs, and then also steroids, because they burn out GABA A receptors. So when you combine a fluoroquinolone with steroids or non-steroidal anti-inflammatories, it can sometimes even permanently burn out the GABA A receptors, where these people will have to become dependent on something like Xanax or Valium, benzos. I have seen several Floxies myself. Floxies are people who have been damaged by fluoroquinolones. And I met many of them. My mother, one of the reasons she's dead today is because the mixture of Vioxx and Cipro, which is a fluoroquinolone and an NSAID. She suffered with 3 brain aneurysms. But for people who have been damaged by the fluoroquinolones, many of them tell me after being on NSAIDs and or steroids with the fluoroquinolone that they have even heard voices and had hallucinations with widespread panic. The FDA is only recognizing fluoroquinolones in combination with NSAIDs and steroids, but what we are learning from many of the Floxy advocates is that you can be given a prescription of steroids a month prior to the treatment with fluoroquinolones and still burn out your GABA A receptors. So many people who become chronically ill and you see elevated glutamate and low GABA, you might want to go back on up to a 20-year history on them if they've taken a fluoroquinolone, because they've been out for a couple of decades here in the U.S. I don't know how long they've been out in Australia, but I know they're going around there now. If you feel your patient has issues with GAD, the best test is the lumbar puncture to test for neurotransmitters. Less accurate, but you can get in the ball field that they're having a problem, or urine, and then blood after. I think I'm done with GAD.
Okay, thanks, Sterling. All right, so we'll just move on to show how this actually works. And you can see here that GABA, as we said, synthesized from glutamate by the enzyme glutamic acid decarboxylase, so that's our GAD. It is stored in the vesicles within the synaptic terminal of neurons. You can see it sitting there in the round circle. And when an action potential comes down the axon and reaches the synaptic terminal, calcium channels are activated, and that results in our calcium influx. And this in turn allows the neurotransmitter within the cell membrane to be released. In this case, it's GABA, which gets released into that synaptic cleft. The GABA diffuses across the synaptic gap and binds to its receptor. And that's what Sterling was just saying. The GABA receptors are GABA A and GABA B. And their activation leads to a hyperpolarization of the postsynaptic membrane and a decrease in the excitability of the cell. That's the mechanism of what we're talking about. And the response is then terminated by desensitization of the receptor and removal of GABA from the synapse into the presynaptic terminal or surrounding glial cells. And that's achieved through specific transporter proteins, and we call those our GABA transporters. And once taken up into a cell, the GABA is taken up into the mitochondria and metabolized, as we said, to succinic acid, and then it enters our citric acid cycle, which we then can use for energy, and then it can be recycled as well, and the whole process starts again. So when we're talking about someone, and I think one of the key things too is to remember that high glutamate can also be very toxic to the brain, and it can go hand in hand with ammonia production. And so it's this excitability of nerves, and I've had patients who say that they feel like they're getting electric shocks inside their body, not outside, but inside their body. And that for me is a really good clue that we're talking about an excess in glutamate receptors, because we've got excess glutamate in the body creating this excitability of nerves. So we were talking about the other day, these are some of the foods that we would expect to see that would be high in glutamate, or at least be excitatory, and so we should avoid these if we suspect that there are issues around the GAD genes. And you'll often see some people will come up with a significant number of these genes to be homozygous. And for me, that's a first thing I would think is, are we seeing any issues with glutamate to GABA conversion? What are they eating? What are some of the things that they've got that perhaps could be contributing to this? And we always think, great, slow cook meats and stocks and things like that, great. Well, not for someone who's got high glutamate levels, because you'll have an increase in this excitatory behavior because of it. And I've seen little kids, when we take away the slow cooked meals and take away some of the things that we would think would be fantastic, they definitely do settle. So if we look at some of the genes, you can see here that they really are associated a lot of the time with quite significant neurological issues, anything from bipolar disorder in children to seizures. And the seizures really relate more to vitamin B6 deficiency and or elevation. And obviously all these receptors work in unison. And as Sterling said, it has also been associated with alcohol dependence because it makes them feel good. And you can see here it's also associated with mild anxiety, heroin dependence in some situations, post-traumatic stress disorder, schizophrenia, and obviously these don't work in isolation. They work in conjunction with some of our other neurotransmitter SNPs. But it's just important to consider, if you're seeing signs and symptoms of perhaps this excitability, have a think about what's going on biochemically with these pathways, and could our glutamate to GABA pathway be significantly affected. So some of the things that we would perhaps think about using if someone does have excess, obviously we want to be checking, for example, alpha-ketoglutarate. We want to be checking B6, which is dependent, this pathway is dependent on B6. But things like theanine, certainly we find this in tea. It does increase levels of GABA within the brain. It also increases production of alpha brainwaves and enhances those GABA-A receptor response. So this may well be, and I'm sure those practitioners who've used theanine before, is that you get a mixed bag of people that either respond or don't. And so really these people that have issues with their GABA-A receptors could quite easily be the ones that react most positively to it. Inositol, it's also a great part of our B group vitamin. Remember before we said that it helps stimulate our poorly sensitive or our overworked serotonin receptors within the brain, but it also helps alleviate anxiety and depression because it does enhance the ability of GABA to bind to the benzodiazepine receptors within the brain. Magnesium, well, we all know magnesium binds to and activates GABA receptors. It's great for pacifying our smooth muscle, helps to relax. And noni is a fruit It's a herb native to French Polynesia, so I don't know if many of you on the other side of the world would know about it, but it's been traditionally used to help with anything to do with the nervous system, and it again binds to GABA receptors within the brain. Chamomile, been around for 1,000 years. It's a great sedative, but it's due to the flavonoid apigenin, and that binds to benzodiazepine and GABA receptors within the brain. St. John's wort, it's a non-selective reuptake inhibitor across all of them, so it's great for not only serotonin but dopamine, but it also may help inhibit the reuptake of GABA. And valerian is known to prevent or reduce the breakdown of GABA within the brain. Kava inhibits noradrenaline uptake, and so it can be a really good one to use again. We've got great Kava products here in Australia, and I'm sure a lot of the Australian practitioners would be well used to using Kava. Ginkgo has been found to increase GABA in the hippocampus region of the brain, and obviously there's a lot of other herbs that we use, like passion flower and skullcap and hops and lemon balm, magnolia bark. They're all well known to have a calming and relaxing effect that will improve mood and give relaxation. We don't know exactly their mode of action, but most of these, they believe, do act on the GABA receptors and so boost GABA levels. Glycine, it's another one of our inhibitory neurotransmitters, and again, it works predominantly on noradrenaline. Potassium is required to help stimulate the release of GABA within the brain, so a deficiency could well increase anxiety, depression, irritability because of it. 5-HTP, we know, is our precursor to serotonin, and serotonin definitely enhances the effects of GABA, so they all work synergistically. All right, so I'm going to pass you over to Cynthia now, who's going to take you through the IgG and IgE section of the presentation. Thank you.
Thank you, Carolyn. I think before we get started with this immune section, it's very complex. I think I want to go over just some general background about the immune system. The immune system is an inherent self-defense system, and it helps our body distinguish between self and non-self molecules. In other words, what to go after or attack and what not to. Different pathways regulate the different immune cells to help differentiate the body's own healthy cells from disease-causing things like bacteria and viruses, parasites, cancer, and those types of things. Now, an antigen is a molecule. Antigens are molecules that are recognized and stimulate the cells of the immune system. They may be different types of molecules within the cells, like proteins, polysaccharides, nucleoproteins. It may also be a whole cell, like a tumor cell or bacteria or virus, things like that. Immunoglobulins is the term that's sometimes used interchangeably with the word antibodies. And so here we can see a slide where we're looking at antibodies. Antibodies are glycoprotein molecules produced by the B lymphocytes and plasma cells in response to an immunogen or after some recognition of some specific type of what's called an epitope on the antigen. Now, these antibodies have a structure that looks like a Y, and you can see in the center here it's a kind of a mock-up. They vary depending on which antibodies we're talking about, but basically there's a light chain and a heavy chain, and then an antigen binding site over on the right-hand side, and then a section called the FC section, which is the bottom, the two legs going down, and that's really the active region. That's the region that binds to neutrophils, eosinophils, macrophages, and the things that we have, like dendritic cells, natural killer cells, to activate the complement system. But I'm not going to go into all the particulars about the chains and all of that, but if you want to do more research, there's a lot out there. But now we're only talking today about IgG and IgA, and IgG, IgA, IgE. But I want to talk a little bit about all of them. IgG makes up approximately 80% of our antibodies. IgM makes up about 13% of our serum antibodies. And it's the first antibody produced during an immune response. So that would be something that you would detect right after, let's say, a bacterial infection. IgA makes up approximately 6% of the serum antibodies, and we associate this with our gut, our nasal passages, areas where the outside world meets the inside world. IgE makes up a very small portion of serum antibodies, but this is the part of the immune system that reacts in things like mast cell activation. It's a histamine response. So antibodies work by doing they have a couple of different mechanisms. One is they neutralize by binding to a bacterial toxin to prevent it from harming the cell. Same with the viruses. Something called agglutination. Antibodies also enhance activities of other defense systems. And again, the activation of the complement system, which we'll talk about in a moment. Now here's a slide that is really just showing you how the pre-B cells develop and the different processes they go through. I'm trying to think about how detailed I want to get here. The B cells are the first cells to reproduce the light change, which can then combine with the micro heavy chains to become expressed as IgM. So the immature B cells differentiate into the mature B cells, which express both IgM and immunoglobulin D, which I didn't really mention, but immunoglobulin D makes up about 2% of the serum antibodies. The early part of the immunoglobulin pathway is antigen independent. Later it becomes antigen dependent, as this is the memory of the immune system when it comes in contact with something again after it's built the antibodies. On contact with a specific antigen via the surface immunoglobulin, the mature B cell becomes activated. Together with the T helper cells, otherwise known as CD4+, and interleukins and cytokines derived from the T cells, the monocytes, the activated B cell matures into a plasma cell to produce these immunoglobulins. You can see at the bottom those little Y shapes. Those are representing the antibodies around the plasma cell. So Sterling, do you want to talk about the IgG SNPs?
Sure, I'll talk about them. Okay. FcGR2A and IgG, that's Fc fragment of low IgG. FcGR2A polymorphism, excuse me, I'm sorry, somebody's knocking on my door. Hold on.
Cynthia, do you want to just say a bit more about the immune system while we're waiting
Carolyn
for
or Cynthia, can you? Yeah, I agree. Is there anything else that Cynthia can add while we're waiting?
We could talk about IgG immunoglobulins, but I want to go back and just mention one thing. The classes are differentiated within the characteristics based on the heavy chain of the antibody. And each chain have different regions. One's called variable and the constant region. And the variable region constitutes the antibody binding region of the molecule to different antigens. And it consists of about 110 amino acids and varies widely among the different different molecules. Now, like I mentioned before, IgG is the primary type of antibodies found in the blood and in the body's fluids. Now, the current variant report, I believe, has I think 2 or 3 IgG SNPs, but the next iteration will have closer to 200. IgG is the workhorse, and it works to control infections through binding to the pathogens. It activates the complement system to eliminate the pathogens. And mutations in IgG often cause a deficiency in its activity. IgG-mediated binding of pathogen causes their immobilization and binding together via something called agglutination. The IgG coating of pathogen surfaces, and I hope I can pronounce this right, known as opsonization, allows their recognition, allows them to be recognized and then ingested by the phagocytic immune cells. IgG activates the classical pathway of the complement system, a cascade of immune protein production that results in the pathogen elimination. IgG also plays a really important role in the antibody-dependent cell-mediated cytotoxicity and intracellular antibody-mediated proteolysis, in which it binds to TRIM21, which is a receptor with high affinity to the IgG antibody. IgG antibodies are generated following class switching and maturation of the antibody response, and therefore participate predominantly in the secondary immune response. IgG is secreted as a monomer that is small in size, allowing it to easily perfuse the tissues. It's the only isotype of the 5 that has receptors to facilitate passage through the human placenta, and this is where a fetus gets their immune system protection in utero. IgA is secreted in breast milk, and residual IgG is absorbed through the placental and provides the neonate with humoral immunity before its own immune system develops. Colostrum, which is the first initial breast milk, contains a high percentage of IgG. This next slide is talking about the complement system. This is a very complicated slide, but basically the complement system is part of the immune system that helps or complements the ability of antibodies and phagocytotic cells to clear the pathogens from an organism. It's part of the innate immune system, which is not adaptable and does not change over the course of an individual's lifetime. However, it can be recruited and brought into action by the adaptive immune system, which we talked about, the B cells. Now, I don't know if Sterling is back yet, or do you want to keep going here?
Yes, I'm back. Sorry about that. I'm back.
Okay, let's go back up.
I can go back and talk about it.
Okay.
FcGR2A polymorphism is correlated with clinical outcome after immunotherapy of neuroblastoma with anti-GD2 antibody and granulocyte macrophage colony stimulating factor. There is an association of FcGR2A with susceptibility to giant cell arteritis. Human resting B lymphocytes entered a state of sustained proliferation when incubated with both the mouse fibroblastic LTK and that had been transfected with the human Fc receptor and monoclonal antibodies to CD40. The risk of giant cell arteritis in HLA-DRB1 positive individuals homozygous for the FcGR2A 131R allele is increased almost 6-fold compared to those with other FcGR2A genotypes. FcGR2A status will also correlate with marrow remission in people with GCA. GSTM3, I pulled out some interesting studies on this with H. pylori, several of them. But there are studies that link glutathione S-transferase Mu3 and IgG titer levels against H. pylori. So one particular study investigated the association between GST polymorphisms and immunoglobulin G titer levels in serum against Helicobacter pylori. Out of 300 healthy subjects seropositive for H. pylori, they analyzed the relationship of GSTM3 and other GSTM polymorphisms. The results showed that GSTM3 polymorphisms are associated with levels of IgG titer in serum against H. pylori. They also suggested that GSTM3 activity is possibly involved in protection against mucosal atrophy caused by H. pylori, as the levels of IgG titer and PGI are linked to mucosal status. Okay, I'm done with the IgG section. All right, IgE, CD14. I found a study on it with peripheral blood mononuclear cells from asthmatic children with the TT genotype at position 159 of the CD14 gene. And that's the one you guys are looking at right now, the particular SNP RSID up there. And the children with the CC genotype, they had an increased risk of having elevated IgE serum levels and higher rates of asthma. Now the DARC gene and IgE, this is called the Duffy antigen receptor for chemokines. DARC is associated with asthma and IgE in 3 populations. So asthma was significantly associated with the RS281-4778 polymorphism in the African, Caribbean, Colombian, and Brazilian families. High total IgE levels were associated with this variant in African and Colombian families. The variant allele was not polymorphic among European Americans. So the DARC gene may not hold weight in somebody who is of European descent, but would hold more weight in people who are African, Brazilian, Caribbean descent. The susceptibility to asthma and atopy among certain population of African descent is influenced by a functional polymorphism in the gene encoding Duffy antigen receptor for chemokines. This genetic variant, which confers resistance to malarial parasitic infection, may also partially explain ethnic differences in morbidity of asthma. All right, FCER1A. FC fragment of IgE, and also called high affinity 1, and then receptor for alpha polypeptide. The immunoglobulin epsilon receptor, known as IgE receptor, is the initiator of the allergic response. When 2 or more high affinity IgE receptors are brought together by an allergen-bound IgE molecules, mediators such as histamine that are responsible for allergy symptoms are released. This receptor is compromised if an alpha subunit, a beta subunit, and 2 gamma subunits. The protein encoded by this gene represents the alpha subunit. So in one study, it was they successfully showed an association between FCER1A variants and serum IgE levels present in cord blood. Small children were independent of environmental stimuli, which seems to be an important genetic input on our understanding of multifactorial etiology of allergic disorders. Authors of this study, Wadiger and Chen, demonstrated 2 common variants, FCER1A, RS24227837, and RS2251746 to be strongly associated with serum IgE levels in general and pediatric populations. The RS2251764 has shown to be a potent regulator of FCER1A expression in mast cells and or basophils. Interleukin 13. Again, IgE antibodies play a crucial role on allergic type 1 reactions. Only IL-4 and IL-13 are able to induce an immunoglobulin isotype switch to IgE in B cells. In one study, people doing the research used an in vitro culture system in which the production of IgE was dependent on endogenously produced interleukin 14 and interleukin 13. And cultures of purified T and B cells from allergic asthma patients and non-atopic controls. T cells were glycolytically stimulated to obtain IL-14 and IL-13 and subsequently IgE secretion. The neutralization of IL-13 led to a significant stronger inhibition of IgE production in the patient group. They observed a higher production of the IL-13 by the patients, while the production of IL-4 was not significantly different. The results that allergic asthma patients produce more IgE in response to exogenous IL-13. All right, now I'm at RAD 50. That one's pretty interesting. Hold on one second. The RAD 50. This one again is in combination with FCER1A will increase IgE. They had in multiple studies the top SNP significantly associated with allergic sensitization on basophils and genome-wide expression profiles indicated an interesting regulatory mechanism of FCER1A expression. Polymorphisms within the RAD50 gene were consistently associated with IgE levels and increased the risk for atopic eczema and asthma. So the RAD50 gene might represent additional factors in combination with FCER1A. All right, SOCS1 is a critical regulator of multiple signaling pathways, including those activated by cytokines that regulate immunoglobulin heavy chain class, switching to IgE. Overall, SOCS1 acts as an inhibitor of IgE expression. So carriers of the T allele of SOCS1 G820T were found to have increased transcriptional activity of the SOCS1 promoter in reporter assays in human B cells. The data suggested that genetic variation in the SOCS1 promoter may affect IgE production. IgE levels increase as levels of SOCS1 decrease. So SOCS1 deficiency is related to elevated IgE in mast cell. Okay. All right. Now the IgA slides. CFH. Levels of urinary complement factor H in patients with IgA nephropathy, that's inflammation of disease of the kidney, are closely associated with this disease activity. Factor H plays a key inhibitory role in the control of activations of alternative pathway complement system. One study showed that there is a predictive value of factor H as a biomarker of renal injury in IgA nephropathy. The levels of urinary factor H when expressed as a ratio of urinary creatinine were significantly higher in those with factor H deposition in the kidney than those without deposition. So levels of urinary factor H may be a useful biomarker to evaluate kidney injury and IgA nephropathy. All right. HLA. In this particular HLA, RS 9271366, it is HLA-DRB1. I need to put that next to it on the variant report and an update probably tonight. But the HLA class 2 histocompatibility antigen, known as DRB1, 11 beta chain, it binds peptides derived from antigens that access the endocyte route of antigen-presenting cells and presents them on the cell surface for recognition by the CD4 T cells. HLA-DRB1 has been associated with IgA deficiency and lupus nephritis. Now, HLA-DPB2 is a major histocompatibility complex class 2 DP beta 2 pseudogene. HLA-DPB2 is related to susceptibility for IgA neuropathy. Now here's a big one, HLA-DQA2. HLA stands for the human leukocyte antigen, and HLA molecules are involved in immune regulation. They bind to small fragments of bacteria and virus and present these to T cells. HLA-DQA2 molecules are particularly efficient in binding and presenting gluten fragments, and that individuals who carry these particular HLA molecules are more prone to develop celiac disease. It is important to remember that the very same HLA molecules that present gluten fragments also are very efficient in presenting fragments of virus and bacteria and are thus of great importance for the body to survive from various infections. This gene is related to IgA deficiency and celiac disease as well as gluten sensitivity. Your HLA type can be called your body type, just as your particular ABO type is your blood type. The prevalence of high IgA antigen antibodies, or HLA, DR, and DQ antigens, are found in trisomies, particularly in children with Down syndrome. They will have a much higher risk of celiac disease. It's basically best if you have someone with Down syndrome to be totally gluten-free. I know it's hard, but that one's really important there. Now, IFIH1. The associations of IFIH1 have been associated with IgA deficiency. And a study in Sweden and Iceland showed that out of 430 affected individuals and 1,090 matched controls, RS1990760 was related to not only type 1 diabetes and lupus, but also IgA deficiency. IGF-1R. Polymorphisms of insulin-like growth factor 1 and IGF-1 receptor contribute to pathologic progression in childhood IgA nephropathy. IRF5, interferon regulatory factor 5 gene, RS47281142, is related to IgA deficiency, multiple sclerosis, pharmacological and clinical outcome of interferon therapy in multiple sclerosis, systemic sclerosis, lupus, schizophrenia, lupus nephritis and IgA nephropathy, inflammatory bowel disease, ulcerative colitis, polymyositis, and dermatomyositis. Sorry, a little tongue-tied with all the itises there. Now, MTC03P1, this gene is also related to IgA nephropathy and lupus. PSMB8 is upregulated in tissue with active IBD lesions and is related to IgA nephropathy. And IBD is inflammatory bowel disease. The TRAF1 gene is also called TNF receptor associated factor 1. This adapter molecule regulates the activation of NF, kappa B, and J and K. It plays a role in the regulation of cell survival and apoptosis. TRAF1 is related to IgA deficiency and primary biliary cirrhosis. And I believe I'm done there.
Okay, let's go to slide 21. I'm going to finish that one up. One thing I wanted to say about the IgA SNPs too, those HLA SNPs are associated with Neanderthal. So people, when they get their 23andMe test results, I don't know if they still do this, but they'll tell you what percentage of Neanderthal genes you have. And they're really talking about these HLA SNPs because they were needed back in the day, but now they're because of our environment, they're not needed, and they tend to be overreacting, like Sterling said with the gluten, presenting gluten molecules, where in the past they were presenting viruses and things that Neanderthal might have been exposed to and needed to have that strong protection. Anyway, that's just an aside. So going back to this IgG pathway and this complement system, like I mentioned, it's what the name says. It complements the immune system. The proteins and the glycoproteins that make up the complement system are synthesized by hepatocytes, so that's in the liver. But a significant amount are also made in tissue macrophages, blood monocytes, and the epithelial cells of the genitourinary tract and the gastrointestinal tract. This is it's typically activated with the immune response, right? So once the antibodies are starting to be produced, the complement system kicks in and helps with sensitizing and being able to identify the pathogen. There's 4 basic functions here. One is called opsonization, which is a way to enhance the phagocytosis of the antigens. Another one is called chemotaxis, which helps attract the macrophages and the neutrophils to destroy the invader. The other one is cell lysis, which is basically the mechanical ability to rupture the membrane of the foreign cells. And then, of course, agglutination, which is where there's a clustering and binding together of pathogens. They're kind of sticking together, and they become a much better target for our immune system to go after. So some of the IgG SNPs, some of the functions here that are associated with FCGR2A, some of the studies are showing an increased risk of gastric cancer for that risk allele A. There's other studies that have to do with rheumatoid arthritis in the Europeans, but not the Asians. The GSTM3, which Sterling mentioned, is associated with the higher risk of Alzheimer's disease. Again, this is, your glutathione S transferase mu3 gene. I'm just looking at my notes here to see if I want to say anything else. The brain-specific glutathione S transferase mu3, the GSTM M3, co-localizes with amyloid beta plaques in Alzheimer's disease. And so there's a link here. A functional polymorphism in the RS7483 may contribute to the decrease of glutathione S transferase mu3 expression in Alzheimer's disease. This particular allele with Alzheimer's disease was significant in women that also had the APOE4 SNP. A significant association was also found in both MCI and Alzheimer's disease subjects with a history of Alzheimer's disease. Let's go to the next slide. IgE. Okay, I'm just going to kind of step back for a minute. When I'm looking at somebody's variant report and I see a lot of SNPs and IgE, I start looking for a kind of a hyperinflammatory response, skin issues, things where they get a pretty significant histamine release. When I'm looking at IgA SNPs, I'm looking for things that have to do with the gut. If they have the IgA plus the FUT2, now we're talking, potentially, autoimmune conditions. But IgA, an overview for IgE here is that this is a type of antibody that's only found in mammals. It has an essential role in type 1 hypersensitivity, which I said before manifests in various allergic diseases such as asthma, sinusitis, some types of sinusitis, rhinitis, some food allergies, urticaria, and atopic dermatitis. IgE plays a pivotal role in responses to allergen, such as anaphylactic type drugs, bee stings, antigen preparations used in desensitization immunotherapy. So again, here you're looking for something, a response that's kind of hyper over the top. Let's say somebody gets, bit by a bee and their throat swells up. This is the IgE part. Mutations in IgE are associated with an IgE deficiency. Although I have to say in some cases the opposite is true. Here's a simplified diagram showing the IgE pathway and the events that lead to the allergy initiation. So let's say the allergen enters the body, which is A. Allergen A enters the body. B. An antigen presenting cell takes up that allergen molecule and presents its epitopes through the MHC2 receptor onto its surface, and there's actually two MHCs. There's MHC1 and MHC2. The activated antigen presenting cell then migrates to the nearest lymph node, which here is shown as C, where it activates T cells that recognize that allergen. They then give the decision for the T cell to differentiate to Th2, to a Th2 cell, D, at the same point. The B cell recognizes the allergen, and through the activated Th2 cell, E, so you see the E right there. The B cell is activated, and at F you see that it's differentiating into the plasma cells, at which point they would actively synthesize the antibodies of the IgE isotope. The IgE antibody that now recognizes the epitopes of the allergen molecule circulates around the body through the lymphatic and cardiovascular system and finally binds to the receptor on the mast or basophil cell. At H, when the allergen re-enters the body at a later time, it binds to the IgE, which is on the cell surface, resulting, in this case, in an aggregation of the receptor, causing the cells to release these preformed mediators. One of these mediators is histamine, which causes the symptoms of allergic inflammation that we know, heat, pain, swelling, redness, itching. Another mediator is interleukin-4, which affects more B cells to differentiate into plasma cells and produce more IgE. And so this vicious kind of feed-forward cycle continues until the immune system stands down. And I know Sterling mentioned this with I think it was the SOCS SNP. Now, some of the risk alleles associated with IgE SNP function are listed here. The risk allele for CD14 is associated with reduced expression and also reduced methylation. Under DARC, the risk allele is C, and this is associated with an increased expression of IgE and a higher risk of asthma and inflammatory conditions. So here's something that's really more associated with an upregulation, so to speak. FCER1A, again, increased IgE level with a higher risk of kind of an over-the-top reaction. And interleukin 13, there's a list here of some of the things this T risk allele is associated with. Increased risk of psoriatic arthritis, increased risk of asthma, reduced DNA methylation with a higher risk of asthma. Here's a couple more of the IgE SNPs with the risk alleles. RAD50, increased IgE level with a higher risk of asthma. So again, we see this over-the-top kind of reaction. Now the regulation, it said that the part of the immune system, the enzyme that we're looking at here that says to the immune system, okay, we're done, stand down, is this SOCS1. So Carolyn's going to walk us through the IgA portion.
Okay, thanks, Cynthia. All right, so IgA or secretory IgA, it's an antibody, as everybody said, that really plays a key role in our mucosal immunity. There's more IgA produced in mucosal linings than pretty well any other type of antibody combined. And we've got approximately 3 to 5 grams secreted into the intestinal lumen every day. That's a lot. And it accumulates to up to 15% of the total immunoglobulin produced in the entire body. There's 2 subclasses, IgA and IgA2, IgA1 and IgA2. And in its secretory form, IgA is our main immunoglobulin in the mucus secretions. So we've got them obviously in tears, saliva, sweat, colostrum, and secretions from the genitourinary and gastrointestinal tract, prostate, and respiratory epithelium. And it is found, as Sterling said, in small amounts in the blood. The secretory component of IgA protects the immunoglobulin from being degraded by our proteolytic enzymes, and so it's the secretory IgA that we know can survive in a harsh gastrointestinal tract environment. And we often see disturbed secretory IgA in our people who have had long-term gastric disturbance. And it's not, we like to see secretory IgA in the gut sitting at about 120, but it's not uncommon for us to see people with either, 20 or 30, or on the other side of the scale, sort of 600 to 800. And so when we're looking at increasing secretory IgA, obviously vitamin A and our good flora is really important, and so is glutamine. But again, we've got to be cautious with our glutamine. And if it's really high, then we know that there's been an overactivation of the immune system. And we've got to look at perhaps what has been affecting it. Is it food sensitivities? Is it the fact that there's such dysbiotic bacteria that it has caused the immune system to go on to high alert? So it does give us some really good indications of what's happening, particularly from a digestional point of view, and our immune system, because we all know that our immune complexes are formed in the gut predominantly. And some research says that could be anywhere between 70 and 80%. And this is quite a complicated little pathway, but really what it's just telling us is that the colonization of the intestine with the bacteria is associated with a really abundant generation of this secretory IgA from our B cells, which are in our gut-associated lymphoid tissues. And when they're secreted into the intestinal lumen, IgA is then what gives us that protection against pathogens. And obviously it has, as I said, a key role in our maintenance of our gut microbiota. And so this really just looks at the pathways of generation of IgA in the gut. And it's quite a good article if you want to read it. It is quite complex, but it just goes to show you how these are formed through the Peyer's patches, the isolated lymphoid follicles, and the lamina propria. And if we look at some of the SNPs here, we've got, as Sterling said, the CFH is associated. This is our complement factor H related protein 1. And that has been associated with IgA nephropathy. And in fact, most of these SNPs, when they are homozygous, can definitely have an effect on nephropathy. HLA, as we said, that is, as Sterling said, gives us an increased risk of ulcerative colitis, Crohn's disease, and SLE, particularly in Caucasian, African, and Asian. And again, remember, there's a lot of differences across the different continents. So we've got to be looking at these and looking at the research and see if we can find, because they're not going to be working in isolation. Obviously, they work with other genes, particularly FOOT2 and some of the others that have more of an impact on gut. And the HLA DPB2, the A is again associated with IgA nephropathy. And the DQA2, we're going to talk about this one a lot more when we get to the celiac disease component. It is also associated with more of a sensitivity towards mold, but here it is systemic sclerosis. And IFIH1C has the link with type 1 diabetes. So I think it's important that we have a look at all of these particular SNPs. And, it is really difficult looking at these and saying, well, what's my overall effect? But I think we've just got to consider these in conjunction with our detailed case history and what are we actually looking for? Is there a susceptibility? Has there been a huge... is this person, have they suffered for many years with gut disturbances? Have they got a family history of gut disturbances? And then that's where we would look at this and say, okay, you have a susceptibility there, so what can we do to support secretory IgA? And here's some more studies that link with inflammatory bowel disease, allergies, nephropathy, and rheumatoid arthritis when it comes to the TRAF1. So if we look at predominantly, we know that vitamin A we get from eggs, cream, liver, kidneys. Obviously, cod liver oil is another one that is very high in vitamin A. Zinc is crucially important for this whole pathway, as is vitamin E, lycopene, our fruits and vegetables, some of our grains, and supplements. We want to support our immune pathway, so there's so many supplements that we know, and herbs indeed that affect our immune pathway. We've got to be very careful. Some people with the BCOM1 gene, and I'm sure Sterling's going to talk about that more when we get to it, but that really affects beta-carotene conversion to vitamin A. And I often find that those people with the BCOM1 homozygous SNPs, and we tend to see a lot of them go together, they really do have a vitamin A deficiency, which is going to affect this whole pathway. So they're going to, on the whole, they will have this inability to convert beta-carotene to vitamin A. So it's not easy for them to do that conversion. So we've got to support that with vitamin A itself. All right, so I think we've got to where we're going to take on some questions, Cynthia and Suzanne.
Okay, so thanks for a great presentation again. We have got one question here at the moment. So while I'm asking this question, if any of the other attendees today have questions, please type them in the question box for us. So the first question that we've got is from Jennifer, and it's for Sterling. Will HLA DQ8 SNPs be available on the report in the future?
Yeah, we're working on getting an entire HLA section out on an autoimmune variant report that we're working day in and day out on. It's just it's been in the works for about 1.5 years, and we're in the last stretch of it. The next couple of months, we should be done with it, and we're going to have an entire HLA section, very detailed.
Excellent. Are there any other questions from anybody? Because we don't have any on the question box at the moment. Hang on, here we go. Thank you, Donna. Okay, so could you please say the name of the gene that converts the beta-carotene to vitamin A again? I couldn't quite hear the letters you said.
Oh, yes. B for Barry, C for cat, O, M for Mary. BCOM1.
BCOM1. So I've also typed that in the chat box for you. So I'll send that.
And we will cover that in the next couple of presentations. I think it's the one after next.
Okay. And any more questions from anyone, please?
Cynthia,
Is there anything that that you any of you want to add? Carolyn, is there anything that you want to add, or Cynthia? Sterling?
No, I think Cynthia was going to add, she wanted to add something with regards the glutamine rather, and how it relates to cancer.
Yeah, I just finished listening to, and I'm sure a lot of you did too, a 9-part series about cancer. And I was an advocate of glutamine, helping heal leaky gut, until I heard this series. And it was frightening, because they were talking about, as everyone knows, that cancer is fueled by sugar, right? So you don't want a person that's undergoing chemotherapy to be eating sugar. Well, the other thing that fuels cancer is glutamate. And so there's controversy now whether or not, if you take glutamine as a supplement, is it converted to glutamate? And if it is converted to glutamate, is it enhancing your potential to develop cancer or to fuel the cancer? Glutamate receptors are not only in the brain, and then there's the controversy about, well, does glutamine to glutamate cross the blood-brain barrier? But there's glutamate receptors all over the body. And so for me, I've taken glutamine out of my protocols, and I've substituted other things. Carolyn mentioned the vitamin A link. When I see somebody with the vitamin A SNP, and I see HLA SNPs, and I see IgA SNPs and FUT2, then I know we're dealing in an area related to autoimmune. So keep that in mind, and don't overlook the vitamin A component.
Yeah, good point. Yeah, and I think this whole issue with regards to glutamate and cancer is a really interesting one, and I know that I have not been using glutamine for a couple of years now, because I know that some people definitely react to it quite adversely if they've got any excitability of nerves or any issues. So I tend to decrease it and just don't use it anymore.
I wanted to add something. When I first initially got out the IgE, IgA, and IgG section, which is going to be much more extensive on the autoimmune variant report we're going to be coming out with, that's probably even bigger than this variant report we have right now. Tim Jackson, Dr. Tim Jackson, is the one who encouraged me to put it out. And we were just talking about this earlier. He is very, very, very well versed in cytokines and IgA, IgG, IgE, and a lot of the other ones like IgM, IgS, which will be on a newer variant report. And he is an admin on the Facebook, MTHFR Facebook wall. His website is drtimjackson.com, and I encourage any of you guys who want to learn more about how cytokines play a role with gut function and all these IgA, IgG, IgE SNPs, because he is very well versed in it.
Okay, Donna is actually saying here, but wouldn't you want a small amount of glutamine for helping the gut's mucosal tissue? Seems like it's about balance and not too much, lots of question marks.
Yes, but for me personally, and for the clients I work with, I'm not willing to risk that anymore.
No,
until I know more.
either.
Until I know more about this.
Okay, and then I've got another question here. What are you using instead of glutamine?
Oh gosh, there's a lot of things you can try. It depends on what their issues are. So there's marshmallow root. There's different kinds of blends you can get for gut support. The one that I like is something called Biotogen from Clear Labs. It does not have glutamine, and it's a good support.
Okay. Andrea says here, interesting you mentioned the effect of high glutamate potentially being toxic to the brain, with people feeling as though they are getting electric shocks within the brain. Could this also be experienced as skin tingling, or have you seen this before? If glutamate levels are elevated, how can these be reduced to avoid that toxicity? If supplementing with GABA, presumably this won't reduce glutamate as it's bypassing that pathway. So who wants to take that?
Cynthia, do you want to start and then I'll...
No, you go ahead.
Okay, so let's take the first part of this question. Just say the first part of that question again, Suzanne.
Sure. Interesting you mentioned the effect of high glutamate potentially being toxic to the brain, with people feeling as though they are having the electric shocks. Could this also be experienced as skin tingling, or have you seen this before? That's the first part.
Okay, so this excitability of nerves can actually be, it can be happening anywhere. So, for example, it doesn't have to be just in the brain. I get some people who they have this almost like not quite cramping, but twitching in the gut. And so when they might not be able to eat because they've just got this constant twitching in the gut. And when you say, is it cramping? No, it's definitely not cramping. It's almost like they say it is a tingling. It's like this constant tingling internally, not so much externally. You know how you can get electric shocks externally. They say, no, it's not that. It's internal tingling. It's internal stimulation, internal shocks, and that can be anywhere: legs, arms, gut, and brain. I guess are the key ones where I would find people would actually experience that. And I think the second part of that question was if the glutamate was elevated, how can you reduce it? Was that?
That's right. How can this be reduced to avoid that toxicity?
Okay, so you've got to work out why, go back to your biochemistry, why would they be getting elevated glutamate levels? So think back on the kynurenine pathway that we talked about the other day. What is stimulating NMDA receptors to the point where we're getting this hyperexcitability? That's what we've got to get to the bottom of. Is it a chronic virus? Is it bacteria? Is it inflammation? So we've got to try and get inflammation down. So you go back to the biochemistry always. That's what I always say to my practitioners. Don't think conditions, think biochemistry. What's going on? So first of all, we have to take away how they're getting this excitability of nerves. GABA, to some degree, will help. Not if you're using pharma GABA, because it doesn't cross the blood-brain barrier. But if you're using GABA that does cross the blood-brain barrier, then that is going to have a very calming effect. Think of our other inhibitory neurotransmitters. Is taurine deficient? Is glycine deficient? Is magnesium deficient? Is B6 deficient? All these things will have an impact on the excitability. Taking out high protein diets in the short term, definitely. And I think a lot of people are making themselves worse by all these paleo style diets, because we are definitely decreasing our glutamine because of it. And they say that roughly with a high protein diet, you will be reducing glutamine levels by 30%. And how many of our patients put themselves on these really high protein diets? And I think it is making it worse. So again, balancing the diet and making it more lower protein, again, high complex carbohydrates, perhaps even supporting with branched chain amino acids, alpha ketoglutarate, B6. That's what I would look at doing.
Yeah, I wanted to add one thing, and we're going to talk about this at the next session, but people that run higher dopamine levels tend to run lower GABA levels, because the body will use GABA to damp down dopamine. So you have to look at the COMT enzyme and see if you can determine whether or not that person is running a higher dopamine levels, because that will deplete GABA.
There's another part to this question, and I'm not sure if it's been answered really. If supplementing with GABA, presumably this won't reduce glutamate as it's bypassing that pathway.
No, it actually will help to reduce the excitability of nerves, definitely. GABA, taurine and glycine will all help if indicated.
Okay, and Scott here says that an alternative to glutamine is slippery elm gruel.
Yes, but I think that's high oxalate. I don't.
Yeah, it is.
I think it's a high oxalate food. So
it is, and so
yeah.
I guess they have to determine, don't they, whether or not oxalates are indeed an issue. And if they're not, great. If they are, then you've just got to be careful.
Or at least make sure that they're getting a supplement with maybe P5P and some peroxidine in it.
Yeah, absolutely.
And also, Donna's just saying here, would you recommend calcium as a supplement?
Not necessarily, because remember that if calcium is going to control the crossing of the opening of the channels for glutamate GABA receptors. So if you're increasing calcium levels too much, you could actually be creating more. So you've got to be careful. I mean, that would have to be a balancing act, and you have to look and see whether it would be too high or too low. But it's calcium that determines the level of glutamate GABA crossing that synaptic cleft.
And I don't recommend giving a mineral like calcium in isolation. I don't do that because of this. I always try to use a blend or calcium magnesium or something like that, but not just calcium.
Okay, there's a few more comments that have come through, which I kind of feel have been answered really. So here, would you want plenty of calcium into the diet or as a supplement with GABA? So I think that you've really just answered that.
Yeah.
Yeah, and yeah, it's just saying here again, just to confirm, I want to make sure that I've covered all of your questions. So thank you for putting them through. Did you say GABA pulls calcium into the cell?
Another way round. Calcium determines the amount of glutamate to GABA. So you remember, I don't know if I can go back to that slide. I'll see if I can, back to the beginning.
Dr. Yasko has a good description of calcium and these receptors. She talks about calcium as bullets from a gun, firing from a gun.
Yeah, exactly. And high calcium can be there it is. So these are calcium channeled pathways. So you see across this synaptic cleft, that is what the calcium regulates the activation of these calcium channels. And this is then going to lead to this neurotransmitter being released. So if there's too much calcium or not enough calcium, then you're going to have this impact of GABA being available to be released into this synaptic cleft, because it'll diffuse across. So I think one of the other things that Amy Yasko always looks at is you've got to be able to make sure that your calcium levels are not too high or too low. So you can check that. I mean, you can check it in the bloods. You can check it. That's probably the best. And you can see corrected calcium and see if it's either too high or too low. And if it is too low, then increasing calcium through foods would certainly, or a calcium magnesium combination would be a good idea, because it is going to help this release of GABA across this synaptic cleft.
Okay, thank you, Carolyn, and thank you everyone for answering the questions, and particularly thank you to the attendees for asking those questions as well. It appears that we don't have any more, she says, and there's something that's come through just as I'm saying that, so let's cover it. Okay, so Jennifer is saying, if someone already has received their report, how do they access any future updates like you have mentioned, e.g. the HLAs?
Oh, that's probably best for Sterling to answer that one.
Well,
yeah.
We're actually going to have a total separate variant report when I do this autoimmune variant report, because there's a few thousand SNPs on it, more than what we have on this one. So it's going to be launched as a totally separate variant report. I have a crew and a team. We're working on it around the clock. When I'm in bed, somebody's working on it. When I'm away, and when they go to bed, I'm working on it, or someone else is. It's that extensive. Updates to this particular variant report that we're doing right now, that is automatically included. As you go on the live one right now, you'll see we're putting videos next to many of the SNPs on this particular report right now. That's all included in this one. But unfortunately, the autoimmune section is so vast and so extensive, and we're trying to stick more with pathways on this variant report, that they're going to have to be separated. They're that big.
Okay, thanks for that, Sterling. So it doesn't seem that there's any more questions. Is there anything that any of you ladies want to add before I close off the webinar?
I just want to add one thing about calcium. Sometimes you have to be careful with taking vitamin D, especially in cases where you know that there's some inflammation going on, because there's controversy about this, but that vitamin D can convert to another form that will release calcium into circulation if it gets too high, calcitriol. And it's measured by, if you have a blood test, you can measure it as vitamin D 125 as opposed to vitamin D 25 to see if you're potentially liberating calcium.
Okay, thank you. And we're getting some thank yous as well from the attendees for the clarification on all of the points that have been raised. So I think now that it's a good time to finish the webinar. I want to just finish once again with the apology about the timings, but we've now got it right. And what we'll do is we'll change the webinar settings so that it reflects the new time. But in addition, we'll also send you a separate email to make sure that everybody does, in fact, get the new timings. So just so that you know, your next webinar is session 8, and that's on neurotransmitters COMT, and that's actually on Thursday, November the 5th at 7:30 for Dallas, and Friday, November the 6th at 12:30 for the Australian audience. So I'd like to